Hello viewers and fellow scientists. This is a video that was put together by some Georgia Tech biology students, and in this video we're going to be talking to you about mutation. Well, before we get into all the meat and potatoes, we're going to give you a brief summary of what goes on in an ecosystem. Please don't get scared by all the terms I'm going to throw at you. Competition is a big part of ecosystems, and competition in an environment is as a result of limited resources. And when the population of an ecosystem increases, competition increases also. In the picture above, we see different species of frogs competing with each other and with turtles. I mean, they're not competing at the moment, but you get the gist. As a result of this competition, one species of frogs moves away. We're just going to refer to this migrating species as poison frogs from now on. This means that the poison frogs would have to occupy their new home, which is called a realized niche, and survive solely on the resources available there. Part of the food resources available to these poison frogs are insects, and some of these contain alkaloids which are quite dangerous when consumed. While the majority of the frogs cannot survive after the consumption of the poisons, genetic variation within the species allows for a few frogs to survive and reproduce after consuming the poisonous new food source, as seen in the blue frog above. Over generations, with the assistance of both natural selection and genetic drift, two more scary terms, I know, the frequency of frogs that could survive and reproduce after consuming the poisonous insects increased, while the frequency of frogs that could not survive and reproduce decreased. Character displacement of this evolutionary adaptation to certain alkaloids allowed poison frogs to evolve resistance to a variety of lethal toxins. As illustrated, natural selection within the poison frog species results in a disruptive selection where the two extreme phenotypes are the ones that are biologically fit in a highly competitive environment. But why do we care about this adaptation? As biologists, we are always looking for improved ways to understand the way species live today. And in order for us to understand how organisms live and exist in the current ecosystem, we have to understand the patterns of evolution that have led them to have higher biological fitness. Once we have a better understanding of how these adaptations came into place, we can use the knowledge from these evolutionary patterns to improve the biological fitness of some of the species we have today. In species like poison frogs, understanding how they ended up having resistance to specific toxins can teach scientists ways to induce resistance to certain chemicals in other species. Who knows, maybe one day we can find a way to make humans resistant to poison. So now, from a more narrowed-down perspective, we're going to take a look at a study that was conducted by some much smarter biologists. Now it's about to get messy. In our focus article written by Rebecca Tarvin and some other biologists, it was hypothesized that poison frogs were resistant to a toxin called epibetadine because of amino acid replacements in the frog. Amino acid replacement, according to Gavin Crooks and Stephen Brenner, refers to when a protein's amino acid sequence is altered by the insertion and deletion of residues and by the replacement of one residue by another. If this hypothesis is accurate, then the absence of amino acid replacements within a poison frog receptor will result in higher levels of sensitivity to epibetadine. To prove this hypothesis, an experiment was conducted by making replacements in humans and in frogs, after which response curves were generated to see how both species reacted. Let's see how that played out. This is a graph that was replicated from our focus research article, and it shows that the concentration curves for the mutant frogs, the black and the amber curves, start before the concentration curve of the wild-type frog, which is the red curve. Don't worry about the other curves, they're not important. But the fact that the curves belonging to the mutants start before the curves belonging to the wild-type means that the mutants were less resistant as a result of the mutation. Here's another replicated graph from our focus article, and we can see that the human mutants were less sensitive to epibetadine than the wild-type receptor. We can see this because the concentration curves for mutants all start after the black curve, which is for the wild-type receptor. This means that the replacements done in the humans were enough to induce epibetadine resistance. So just in conclusion, to wrap up all we've just shown you, the scientists were able to show that the poison frogs with amino acid replacements show changed levels of sensitivity to epibetadine compared to wild-type receptors. This result provides strong evidence that amino acid replacements is the proximal reason for how the species of frogs evolved resistance to epibetadine. I know mutation doesn't play a direct role in picking which species gets to survive longer, but it sure as hell does play an important one. Other factors such as competition, weather conditions, and predators all play essential roles as well, and a combination of two or more of these may be why toxin-resistant species exist today. The evolution of toxin resistance happens over time in response to continuous exposure. The pattern of resistance in this experiment has allowed scientists to track the evolutionary history of toxin exposure in different species, and I know there are more scientists out there working on ways to improve the knowledge we have so far. Yeah, so finally we're done. So please share this video with your friends if you found it helpful, and don't forget to like it too. Please like it for me though.